So, very good morning to everyone. And it gives me immense pleasure to welcome you to the one-day national webinar on integrative therapeutic innovations, WNT pathway, nanovesicular systems, and drug discovery by the Department of Pharmaceutics under the aegis of Internal Quality Assurance Cell at MSG COPER. It gives me pleasure, Shauna Kulkarni, to introduce you about our prestigious Gokhale Education Society. So the Gokhale Education Society was established on the 19th of February 1918 by Principal T.A. Kulkarni, who was a great social worker on the occasion of the third death anniversary of Namdar Gopal Krishna Gokhale, who was a spiritual guru of Mahatma Gandhi. The society has completed around 106 years of meaningful existence and today has more than 140 units spread over three zones of Mumbai, Nashik, Thane Palghar, and is serving around 1.2 lakh students. Previously, Gokhale Education Society was led by late Principal S.B. Pandit, who was the president of the society and a great visionary and an eminent personality. Late Sir M.S. Gosavi, Sir Dr. M.S. Gosavi, who was the Secretary and Director General, who rendered invaluable service of 65 years in the field of education. At present, Dr. Mrs. D.P. Deshpande, who is the secretary, is playing a crucial role in the overall progression of Gokhale Education Society. So about our college, which is Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, has been founded in the year 2012. So presently, this college offers M.Pharm in Pharmaceutics, Masters of Pharmacy in Pharmaceutical Quality Assurance program with an intake of 15 seats each. The B.Pharm program is permanently affiliated to the Savitribai Phule Pune University with an intake of 100 seats, and the D.Pharm program is affiliated to MSBTE Mumbai with an intake of 60 seats. Accredited by NAC for a period of 5 years, this college has a state-of-the-art infrastructure and has a lush green surrounding, which provides favorable milieu for students to grow not only as a good pharmacist, but also as a world-class citizen. College has an experienced and qualified staff with academic excellence. The college maintains a tradition of good academic results and organizes various activities including conferences, seminar, student faculty development programs, workshops for overall development of students and faculty to bridge the gap of academia and industry. So I'd like to welcome you all to this one-day national webinar, and we'll be having three sessions today. The first session would be by Mr. Sumit Shinde, sir, who is an assistant professor at Sarojini College of Pharmacy, Shivaji University, Kolhapur. Then the second session will be by Mr. Saurav Suryavanshi, who is the prestigious alumni of our college and who is currently in the Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, which is ICT, Mumbai. Then the third session will be by Dr. Suman Manadhar, sir, who is a medical reviewer at Cactus Life Sciences. All the three resource persons will be speaking on very nice and very interesting and intuitive topics, with the session 1 being on emerging trends in drug discovery and translational research. The session 2 will be on nanovascular drug delivery systems, pioneering the future of precision therapeutics, and the third session will be on deciphering the role of WNT signaling pathway in Alzheimer's disease, mechanistic insights and therapeutic implications. So I welcome you all to this webinar, and I hope you make the best of this webinar. Thank you very much.
A very good morning and warm welcome to all the participants for today's one-day national level webinar, Integrative Therapeutic Innovations, WNT Pathway, Nano-Vesicular System and Drug Discovery under the aegis of IQAC. I am Dr. Ashwini Sherke. Welcome you all. It is a great honor and privilege for me to introduce today's eminent speaker, Mr. Sumit Shinde, sir. Sir has completed his master's in pharmaceutical chemistry from Bharati Vidyapeeth College of Pharmacy, Kolhapur, Maharashtra. Sir is pursuing PhD in pharmaceutical sciences at Ganpat University, Mehsana, Gujarat. His area of interest, the study of mutation cancer protein and identification of novel lead molecule against mutated protein target using the computer-aided drug design and discovery methods. Sir has total work experience, teaching experience more than 4.5 years. He is working as an assistant professor, Department of Pharmaceutical Chemistry in Sarojini College of Pharmacy from Shivaji University, Kolhapur. Teaching experience, sir is having teaching experience at Shivaji University and Dr. Babasaheb Ambedkar Technology University, Dibatu. Sir also has an industrial experience more than 6 years in Analytical Research and Development Center, Macleod from the Macleod Pharmaceuticals, Daman, then Intas Pharmaceuticals, Ahmedabad, then Vales Pharmaceuticals from Goa. Sir also has contributed research in academics by guiding the 20 bachelor students in their research projects, then by providing the research guidance to interdisciplinary researchers in computational chemistry, analytical chemistry. Sir also attended more than 20 faculty development programs, workshops, webinars, seminars, and conferences across the India. So we are having a great privilege that we are having Mr. Sumit Shinde sir with us for today's first session. So welcome you, sir. Topic of the sir is emerging trends in drug discovery and translational research. So
Okay.
okay,
Good
sir.
morning, all of
So
you.
you can continue, sir.
Okay, good morning, all of you. Thank for my
Good morning,
lovely
sir.
introduction.
Thank you, sir.
Thank you for program coordinator, Mr. Umesh Bhambre, sir, to give me a wonderful opportunity for presenting my research area or my work. Now, can I share about my slide? That is today we are the deliver the lecture on the emergency trade in the drug discovery and translation research. Okay. Now, firstly, we are discussion about drug discovery. Now, it is a drug discovery. It is a process by identification of the new chemical entity or biological molecule. They have the potential to treat to these cure diseases. And it is another part is drug development. Drug development is involved taking this potential treatment throughout the rigorously testing of and regulatory approval to the final make to the available in clinic for the patient any treatment. Okay, this process is crucial for advancing medical treatment to improving patient outcome and addressing unmet to the medical need. Now, firstly, they are the introduction part. They are the drug discovery. Basically, scope of the drug discovery mainly having there are the various parameter that is a say it is a creation or screening of the new entity or chemical molecule. They are the molecule weight is less than 500 daltons, and second is the biological discovery by the molecule weight more than 500 daltons. They are the like that example is monoclonal antibiotics vaccine. They are the method for modification of the genes by using the gene therapeutics, and they are the discover about the new novel drug for these cancer and cardiovascular diseases to rare genetic disorder. It is a developed to the safe, effective treatment of the diseases, addressing to these medical treatment for using the worldwide market. Now, I focus upon my research work. They are the drug resistance, mainly involving into a previously drug that can be shown into a many side effect. I focus upon this drug discovery. They can be improve existing therapeutical side effects, efficacy, or toxicity study for to enhancing patient quality of the life to treatment of outcome. Now they are the firstly discuss about they are the exactly process for drug discovery and development. They are the two part, drug discovery and drug development. Drug discovery phase they have required 3 to 5 years. They can be discussed about discovery about a small leads. In 10,000 molecule can be screened, they can be given only one molecule. Now the various parameter that is the first is target identification, target validation, compound screening and lead discovery. Then the molecule move toward preclinical development. After completion of drug discovery phase, they can be moved towards drug development phase, like that preclinical development, then clinical development like that, phase 0, phase 1, phase 2, phase 3, and they can be approved for the new and chemical entity to into a market by using a regulatory as per the country norms. Now, overall, this process is required for 10 to 15 years. This struggle for cost of the traditional drug delivery or development process. Now, 10 to 15 years is required, and they are the most important point for they are required 1 to 2 billions cost for this process. It is a huge amount of investment in pharmaceutical industry. Now, they are this scene about share about the fundamental challenge in traditional drug discovery. That is the first challenge is biological complexity. Second challenge is heat identification hurdle. Third challenge is undruggable target. Fourth challenge is limited returns on investment. That the low probability to success means it is a successful drug, means generates sufficient revenue to the cost of numerous failure program. Now, I discuss about it is a major gap between discovery and delivery. Now, the persistent struggle with protect timeless exorbitant cost, high failure rate created widen gap between the fundamental discoveries and the delivery market therapies. This systematically inefficiency directly drive to the imperative for descriptive innovation of drug discovery and development. Now, they have discussion about the evaluation by artificial intelligence based discovery in paradigm shifted. In pharmaceutical industry, they undergoing profound transformation to moving for conventional drug discovery focused to the empirical method toward the artificial intelligence drive to the paradigm. It is a mainly fueled towards the exponential growth of biological and chemical data. There can be remarkable advancement to the computational intelligence. It need to the more efficient drug development process. Next slide, they can be seen about machine learning. Okay, machine learning, it is one of the type artificial intelligence that machine learning and deep learning process have become indispensable for overcoming traditional drug delivery challenges. First challenge is analyze vast chemical database, identify potential therapeutic targets, then predicting drug efficacy and unpredictable speed, then process divert data type beyond to the conventional compound. Okay, they have used for machine learning technique in the architecture like that graphical neural networks, that is a GNN, and recurrent neural network, that is a RNN. Okay, now we have the discussion about is artificial intelligence is unique capability in drug discovery, in that they are the reasoning behind with knowledge, high throughput hypothesis, and they are the augmenting human expertise. Now we are the discuss about first they are the application of AI, artificial intelligence in drug discovery. That is the first application for protein target identification, target validation. Second is lead compound generation. Third is they have absorption, distribution, metabolism, excretion, and toxicity data prediction. Then drug repurposing, then molecular dynamic stimulation, and they are the lead optimization. Okay. Now we are the seen about they are the major pharma industry in onward. They can be moved towards focus upon this part. The AI is used for accelerated for process of drug discovery, for automating and optimizing various stages, rapidly process massive volume of the biological and chemical data. They are the quickly validated biological target. They identify potential drug candidate, the fraction between traditional time. This process is required for in traditional system they have required for 5 to 6 years for one chemical entity discovery. Now in that AI is it is going to show crucible job just 1 year, single 1 year, they can be completed overall the study. Now, they are the main focus upon cost reduction. They are the automation mainly can be reduced into a screening time, reduced into a extensive and cost laboratory equipment. They are not required to the any synthetic chemical moiety, or it is a not used to the multiple stage synthesis. Now they have the filter a huge amount of data to the convert it to a small database. Okay, this small database is required for it is a surety for giving a 100% a positive result. Okay, then faster market entry to shorten development cycle leads to earlier market entry of the new drug to increases returns or investment can be reduces overall cost. Now, AI can be predicted by the approach of that is a structure activity relationship modeling. Then they have the QSAR modeling. Then they have the QSPR modeling, that is quantity structure property relationship. They are the seen about deep learning, or it is a generation of screening about toxicity of compound. Now, and the last part, that is a multiple endpoint for the screening. Now, we have seen about exactly present challenge in drug discovery, that is the data quality accessibility. Second is model interpretability. Third is ethical and regulatory concerns. Fourth is resource and requirement. The positive data, first that is data quality and accessibility, they have seen about. They have the required positive data for structurally, and they are the incomplete data can be mainly involving into this process is negative result. Okay. Now, they are the overall this slide. It is a present challenges occur into a drug discovery. Now it is introduction part regarding to the computer aided drug design in drug discovery. That is a various step. That is a target identification, high throughput screening. That is the high throughput screening. There are the two type drug design: structure based drug design, ligand based drug design. In that mainly occur into a various step. That is a drug design based upon the known protein, unknown protein. The target is not having the target is got for further the study about the drug design. And they are the de novo design, then library design by using the high throughput screening. It is a computer based technology. They can be give towards move toward the lead optimization or hit optimization. Then further they can be give a new drugs or chemical entity. I told about that the one of the drug out of the screening of 10,000 molecule, they can be give only one drug. Okay, there the huge amount of database to screening about, or it is a diamond like one drug. Then there are these screening upon absorption, distribution, metabolism, excretion, and toxicity study. They are the follow to the target discovery, follow to the drug discovery, then preclinical research, then clinical research or trials, then drug manufacturing, and then go toward the approval for the market. Okay, this is the various phase to the scene about drug discovery. Now, total layout in AI in drug discovery. First, they can be, it is a raw material, that is the structure and protein structure. Structure, they can be got into a Unichem, Uniports. They are the PubChem database. They are the drug bank, CHML database, and protein structure they can be got in rcsp.com sites. Uniports, the protein sequence they got a Uniport web server. Now, this database can be generated to set for QSR modeling, that is the training set and test set, depend upon the chemical inhibitor structures, and they are the targeted proteins. Now, they can be data represent that is in the form of smile format. They overall the scene about the upper corner, they have seen about benzene ring. The benzene ring, they have one of the smile structure. And a smile structure, they can be got into a molecular descriptor, or it is a structural properties. Then right side corner, they have discussion about they are the encoding or decoding. They are the replacement of the amino acid. They can be generate a new protein sequence and they can be validated. Then model, after the development of the new protein molecule, they can be validated by using a various server. That is, Save server is mostly used for web server for protein sequence validation. Then there is a result and discussion. They can be predict multiple targeted proteins, and they can be predicted new chemical entities. Okay, then model optimize by using they are the based upon their particular rules. Then they have this slide is this describe about the historical development of machine learning in molecular locking study. Exactly, they are the various stage. In 1943, they are the machine learning first neural networks to the model toward the move towards 3D printing in pharmaceutics. Now 2022 or 20, they are the machine learning is introduced to the 3D printing in pharmaceutical industry. Now I focus upon my research area on the title of the in silico approach to the screening of ABL kinase protein by using homology modeling technique. It is a firstly seen about for the introduction part. That is, what is the ABL gene? ABL gene is a pronto-oncogene that coded with enzyme tyrosine kinase. This seen about that is a blue-like structure and green-like structure. Now, this is the translocation gene between chromosomal 22 and chromosomal 9. They can be lead to the philopedia chromosome. That is a BCR ABL. Now, this lower side image is clarify about generation of this gene for the modification of the cancer cell by mechanism of cell adhesion, cell division, stress response reaction, cellular diffraction. ABL is family of non-receptor tyrosine kinase, that is ABL1 and ABL2. Now, the previously marketed drug, they can be attack a particular target, that is a ATP binding site. There are the various marketed drug they have seen about right hand side corner, and one of the drug is asciminib. This is a bind with merostral pocket target. There is allosteric binding site. Then I focus upon one of my objective, that is a homology modeling. Homology modeling, it's a firstly seen about the introduction. It is a has a mature into important technique for structural biology to significantly contribute to the narrowing gap between known protein structure and experimental determined structure. They also seen about overlapping study between previously template structure and next query structure. This upper screen or image, they can be seen about sequence of amino acid, can be a little bit modification between amino acid sequence. They can be got using by through homology modeling. Now this modeling further, why it is choose of this topic in research, because they have seen about drug resistance. Previously, 8 drugs of ABL kinase inhibitor. They also seen about drug resistance may be occur into a particular number of the amino acid. They are the resistance mainly occur. They have also seen about red like number, that is a it is a amino acid and they are the number. That is the T315 means it is a tyrosine 315 amino acid. Number of amino acid is 315. Overall, the resistance mainly involved into a previously drug and a pocket interacting amino acid. They also literature survey, they can be got various amino acid. They mainly involving into a drug resistance. In this drug resistance, the drug like that immediate, it does not show biological activity because of this point of drug resistance. Now focus upon drug resistance in the form of they are the mutation of protein sequence. Now, what is the problem statement? On the crystallography study of imetinib is complex with the kinase domain for ABL1 revealed that the drug bind to the catalytically inactive conformation of the enzyme. This study is also identical critical residue within the ATP binding site that is form of direct hydrogen bond and the process between the van der Waals force of interaction with the imetinib drug. This residue at which mutation are associated with strongly resistance to give tyrosine kinase inhibitor are indicated in a red line structure. Those associated with lesser degree of resistance can listed in orange. Both these T315, that is tyrosine 315 amino acid and glutamic acid 2,5 amino acid, the mutation to lead to the increases their IC50 value of for ponarinib. Now, on the basis of literature survey, they can be shown into. Then I focus upon major objective in current research work, that is designing Abel kinase inhibitor based upon the molecule docking study to reduce the existing side effects or toxicity and give a positive binding result or biological activity. On the basis of molecule docking study to screening about one of the protein 40WP, PDP is 40WP, lead to molecule interact with a specific targeted macromolecule. The binding site is associated with current Abel kinase inhibitor for the use to improving specificity and identify ligand help to the eliminating or reducing adverse effect to develop Abel kinase inhibition therapy. To developing such simple, cost-effective, and specific ligand in need of today's world. Now, they are the methodology for using for new chemical entity discover. That is the first objective, that is a building of mutation model. Second is objective, that is a pharmaco-based virtual screening, high throughput screening. They have used shape-based screening. Then they are the molecule docking study. Then they are the pharmacokinetic study, that is a create a toxicity profile. And then they can be shown in molecular dynamic stimulation. Now, today they focus upon first objective. This is build up mutation model. My first objective, build up homology modeling. Now, this previously protein sequence they can be given into Uniport's web server. This protein sequence further modify into a particular amino acid replaced with mutation, that is the second amino acid, and they created two mutation model. On the basis of two mutation model, they can be give one of the Swiss models web server. The model can be generated, this software, and this software can be created into a cumin ratio. They can be create models. They can be evaluated into a further study. Now, this is a scene about the selection of protein by using the rcsb.com, the PDB, this modification of proteins, then model building, then evaluation model, and then model give a positive result. They can be used to the further study. Why positive results? Because evaluation of each and every model is necessary for moving toward the molecule docking study. Now, molecule docking study by using this coordination software for the study by determining to this run our two-side study, that is a structure-based drug design or ligand-based drug design for first preparation of protein. Okay, they are the previously protein and mutation protein. In the using of this coordination software, this same process can be happened into a development of proteins. Then at a time, they are the extraction or a 3D structure to the ligand preparation. Then further, they are the ligand and receptors docking. They give a particular leads molecule. Now, this comparative docking analysis of finally X-ray ligands marketed drug to screening of molecule in homology modeling. Now, I move towards a basic result and discussion. This scene about upper line left corner, this is a black background. This is a structure of a previously protein, that is a two lobe, A lobe and B lobe. That is a two chain A and B. Now I focus upon B chain, and they are the modification between this one of the T315 I chain, or it is a previously model. Now this modify this protein structure to give a new mutation structure model. Now the screen is clarify about slide. This previously protein and mutation protein, they can be superimposable each other They give into a right hand side corner image. They give lower side corner image because they are the match to the give 100% sequence identity by using Swiss model, and further they can be study about by using the save server. Now this image is clarify about this overlap the two sequence of the amino acid. They can be seen about this amino acid sequence in ribbon like structure. They have this seen about alpha lobe and beta lobe. Now this previously protein code 40WP and new generated model T31πL, T31πM, and wild type of the protein that is used to the 7W7X. Now overlapping they possible to the generation of the same model. Okay, this model can be evaluated by using the save server. This is shown into a Ramchandran plot for using for homology modeling evolution. Okay, they have contained 4 quadrant in the graph of pi and psi ratio. There are the various region: allow region, disallow region, flavoring region. Now I research work, they have got into a some molecule, they can be moved towards a cinnabar flavoring region. Now they are the seen about the 4 graph is clarify about they are the documentally seen about they are the sequence identity show in more than 90%. Okay, this graph is clearly this region between the red like structure. This is a red point that is a molecule is majorly molecule or amino acid. They can be flavored to this previously amino acid. Now it is a one of the study that is a pharmacophore study, ligand based drug design. I got a positive result between the got depend upon the functional group, hydrophobic site, lipophilic site, hydrophilic site, and they are the upper lying black background structure that is a bunch of zinc databases got. Total they have above 4,000 molecule they can be matched with once. This structure is clarified about this is a bunch of the structural. Now they can be given into 3D pharmacophore for given into a particularly pocket mapping. Now further move towards on the basis of previously docking molecule or protein. Now this slide is clarify about this is a molecule for zinc I have given. They can be analyzed or modify or dock with various protein that is the previously molecule, then mutational protein that is a model 315L, model 315M, and wild type of the model 7W7X. They are the overall interaction they can be seen about a positive they can be seen about result. Now I discuss about that is the interaction between hydrophilic interaction of the amino acid tyrosine 2pi3 mainly involving into a exactly between the protein and receptor structure, that is the shown into a hydrophobic interaction, and they got a result of XP docking score for minus 11.06. It is a score between the mutational one of the model and a lead molecule. Same lead molecule can be interact previously protein. They can be got minus 9.171 result. Now this study further mutation model that is a T31πM protein and the same lead can be docked together and they can be got minus 10.604 a result. This overall this slide it is clarify about this is the scene between after the modification about or homology modeling they can be give successive result in the molecule docking interaction between these the new leads toward the mutation model. I have got a positive result for required for. Now this molecule is zinc 00030068884. It is used for further molecular dynamics stimulation. Now that is conclude that this work, the structure of the PDB ID 40WP is potential target of Abel kinase protein was modified, evaluated using the various validation method. This protein is conserved active site like that ATP binding site. There is a need to export selective tyrosine kinase inhibitor against the mutated BCR Abel complex protein to efficient treatment of chronic myeloid leukemia. Okay. This is a reference for using for this research work. Okay, thanks to all. Thanks to program coordinator. Thanks to principal sir and organizing committee sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, Nasik. And thanks to all faculty member. Any question? My email ID is any question they have to send this my email ID Shinde632@gmail.com. Thanks to all. Any question for audience?
If participants are having any question, you can put your questions in chat box. Sir, okay, so I think there are no questions. I conclude today's session with it's very great honor that we are having Mr. Sumit Shinde sir for today's session. Sir has elaborated very well on emerging trends in drug discovery and translational research. Sir also elaborated the drug discovery, scope of drug discovery, various development processes in drug discovery in very elucidated manner. Sir also enlightened the fundamental challenges in traditional drug discovery. So I hope all the participants will get benefited in their research work from today's session. So thank you very much, sir, for giving your valuable time for us from your busy schedule. Sir, please accept our certificate of appreciation.
Thank you to all.
Thank you very much, sir. Camera.